Dear students, today I'm going to discuss about the topic precipitation reaction. Precipitation reaction is also a particular type of antigen-antibody reactions where both the antigens and the antibodies are complex. They are actually the complex of amino acids, proteins, and also have positive and negative polar groups distributed over their surfaces in specific but reciprocal patterns. When an antigen and corresponding antibody molecules are mixed, then due to presence of these polar groups, there should be electrical attraction between the different type of groups, means positive and negative polar groups, and there may be repulsion between the same charge group takes place, and this particular reactions between the two different type of polar groups between the antigens and the antibodies results in orientation of the corresponding antigen and antibody molecules with respect to their molecular forms. Here in this picture you can see here these are the antigens, yellow spots, and the green Y-shape, these are the antibodies. So first you can see both the type of molecules, the proteins are present in their solution, so they are guiding themselves towards their counterpart. So here you can see that the orientation took place between the corresponding antigen and antibody molecules with respect to their molecular forms and electrical charges, so that an absolute feed is obtained. Means molecules, means antigen will interact with the antibody. So this particular type of reaction that is to be discussed today, the precipitation reaction, involves the reaction of soluble antigens with immunoglobulin G or immunoglobulin M antibodies to form large interlocking molecular aggregates that is called the lattice. Precipitation reactions occur in two distinct stages. The first stage, uh, where the antigens and the antibodies rapidly form small antigen antibody complexes. Here you can see they rapidly form small antigen antibody complexes. This interaction occurs within few seconds, and it is followed by slower reaction. Here you can see slower reaction which may take some minutes to hours, in which the antigen antibody complexes form some lattice. Here you can see they are forming the lattice. So one by one, they are just interlinking with each other. So this is the lattice formation that precipitate from the solution. So due to formation of the lattice, now their molecular weight will be larger. So now they can be soluble here. So they will precipitate. So this is how the precipitation reaction took place. So here, through this picture, we can say that in this picture you can see that this precipitation method include different type of process. So in this phase, this is the highest one where this lattice is formed. There you can see the most precipitation. And this phase, this green phase is known as zone of antigen antibody excess. And this is the antibody excess. And this one, this blue zone, this is the zone of equivalence, means equivalent amount of antigen antibodies present there, so they are forming the lattice. And this one, this orange one, this is the zone of antigen excess. So we have three zones. One is the first one is the antibody excess, then equivalent antibody of antigen and antibody both are present there. This is zone of equivalence. And the orange one, the last one, this is zone of antigen excess. So in both the two zones, antibody excess and antigen excess zones, we cannot see maximum amount of precipitation. So maximum amount of precipitation occurs at the zone of equivalence. This precipitation method can be of three types. What are the types? So first one is a double diffusion. So, double diffusion is a qualitative gel technique that determines the relationship between antigen and antibody. The second one is radial immune diffusion. This is a semi-quantitative test of proteins by gel diffusion using antibody incorporated in agar. And the third one is electroimmunodiffusion. Here you can see the variation of double immune diffusion method reaction that uses an electric current that enhances the mobility of the reactant towards each other. So precipitation reactions are less sensitive than the agglutination reaction we have already discussed, but this remains a gold standard in serological techniques because this particular technique is very much important. This is the gold standard in serological techniques because most commonly used methods are the Octilon test, where we can see that this particular test is based on double immunodiffusion and named after the Swedish physician who invented it. Second one is Mancini method. It is based on single radial immune diffusion. So the following types, here you can see that the double immune diffusion technique, three basic reaction patterns result from the relationship between antigen and antibodies. The first is the identity. The second one is the non-identity. And the third one is the partial identity. In Mancini method, you can see that this particular method results in precipitating ring formation. We discuss and later on we will discuss the ring formation, ring test on a thin agarose layer, and the diameter of the ring correlates with the concentration of protein in the precipitate. So now we are going to discuss about the precipitation reaction. The flocculation reaction is when it will just flux over the solution. So the ring test, this particular test is used for the detection of antigens. It's very simple by using this test. So in this particular test, a layer of antigen is formed over a layer of serum in a very narrow tube, and the line of precipitate is observed at the junction of the two clear fluids. You see two clear fluids here in this picture. You can see here this black one. This is the soluble antigen, and there is a line of precipitation, and this is the red one is the antibody. Here you can see these yellow particles are the antigen, soluble antigen. Here the green one, they are the antibodies, IgG or IgM. And this is the zone of equivalence where we can see the precipitate of the ring formation. Here in this cone tube, you can see here the formation of a ring, this yellow ring formation between the two clear solution. So this particular test in C-reactive protein test, in Ascoli's thermoprecipitating test, in diagnosis of anthrax, and in typical streptococci are some of the important application of this particular test. So detection of adulteration of food also, foodstuff is also done by using this ring test. Next test is the slide test. Here in slide test, like we have already studied Vidal test in our agglutination reaction, here in precipitation reaction. Also in case of VDRL test for syphilis disease. This is a slide, and in this particular test, a drop of each of the antigen and the antiserum are added on a slide. Then both antigen and antiserum are mixed by secking, and the reaction is observed, which is forming some floccules or not. Whether floccules are formed, then we can say this is positive. So in case of VDRL test is a particular example of slide test. Next one is a tube test. Likewise, Vidal test we have performed tube test. So VD as a slide test is a qualitative test, not a quantitative test. A tube test, we can see both semi-quantitative and quantitative tests. So in this test, a fixed quantity of antitoxin is added in the tubes. Then serial dilutions of toxoids or toxins are added to these tubes. The amount of toxoid or toxin which flocculates optimally with 1 unit of antitoxin is known as a LF dose. And one of the examples of this tube is Khan test for syphilis. We can use for VDRL test. So this is for the quantification, so quantitative test for syphilis or VDRL test. Next one is immunodiffusion. So immunodiffusion is precipitation in a gel. So this particular technique is very important because this particular method is of precipitation reaction is observable in the form of distinct band, which is more stable and if necessary can be stained for preservation. So this ring test these are not stable, but here in case of immune diffusion, as we are doing this test in a gel, so this particular test is very stable and we can stain it for better visualization and for preservation also. So here the number of different antigens in the reacting mixture can be readily visualized as each antigen antibody reaction gives rise to a line of precipitation. Immunodiffusion also indicates to identify means the three different types, means whether identity, non-identity, or partial identity is there. So cross reactions between the between different antigens can also be visualized through this particular type of reaction. So immunodiffusion is usually performed over a soft agar. Here the agar percentage is 1% or agarose gel. So precipitation in gel is more advantageous than the precipitation in liquid medium because it is stable. It is stable. So and there are different types of immunodiffusion tests. First one to discuss is the Odin procedure, that is a single diffusion in one dimension. Here, the agar gel having antibody is taken in a tube. An antigen solution is layered over it. So the antigen diffuses towards downwardly, and wherever it reaches the optimum concentration with antibody, a line of precipitation is formed. The number of bands indicate the number of different type of antigen present there. In case of double diffusion in one dimension, that is Oakley-Foltroff procedure, here you can see that in this particular test tube, means in a test tube, antibody is incorporated in agar gel, and above this layer, a column of plain agar is placed, in which in turn is superimposed with antigen. So either as a liquid or incorporated in agar, the antigen antibody complex will form, and this will show as a band of precipitation, a band of precipitate, where they meet at optimum proportion by moving towards each other through the intervening agar. Here we are using some intervening agar, normal agar, over the layer layer of the agar gel. So this intervening agar is responsible for particular direction. This is known as double diffusion. So next one, the third one, this is a single diffusion in two dimensions, that is called radial diffusion. Here you can see that a slight, but generally a petri dish is used for this method. So agar gel is poured on a slide or maybe a petri plate, and antiserum is then incorporated in the agar. So the well were prepared on the agar surface, and the antigen is added, poured into the well. So bands of precipitate of rings are formed around the wells, and the concentration of antigen is estimated by measuring the diameter of the ring. The second one is double diffusion, that is known as Oakley method. So here, agar gel is poured on a slide of petri plate, like the first method, that is radial diffusion, immunodiffusion. So at different vials are prepared on surrounding and one at a center on the agar. So same type of wells to be prepared, one at the center, and maybe 3 or 4 surrounding the center well. So in the central well, we are adding antiserum, and in the different antigens at the surrounding wells. So if in observation of the two precipitating bands formed totally combined, then the pattern is known as a reaction of identity. So this indicates that the antigen in the adjacent well are identical. If the observation of the precipitating band forms separately and cross each other, then we can say the pattern is a reaction of non-identity. So this indicates that means the antigens are non-identical. So this indicates that the unrelated antigens are present. So means on non-identical antigens are present. So if in observation precipitating bands fuse with form a spar-like projection, in this case, the antigen are cross-reacting. So this is known as a reaction of partial identity. So 3 different type of identity, whether it's a identity, non-identity, or partial identity, we can detect using O'Connell process. So for the diagnosis of smallpox, this method is routinely used in the laboratory. Next one is immunoelectrophoresis. In this particular process, electrical field is applied. So it's an electrochemical process uh, in which colloidal particles, that is the suspended particles, or the macromolecules which having a net electric charge travel in a solution of agar or agar gel under the influence of a current or electric field. That is why it is known as electrophoresis. So here you can see that a characteristic of living cells in suspension are biological compounds such as protein or antigen in solution or in agar gel. Uh, when they are placed in an electric field, they travel to the positive or negative electrode. So depending on the charge of the substance, so positively charged substance travel to the cathode or the negative electron, while the negatively charged one will go to the anode or the positive electron. And this movement is known as electrophoretic mobility. So we are going to measure this electrophoretic mobility. So when electrophoresis is applied to study antigen-antibody reactions, so it is called immunoelectrophoresis. And when a fluid containing antigen in the form of proteins is placed in a well which was prepared in a thin layer of buffer agar. So if an electric current is applied to this, the antigen will travel and distributed to in separate spots along a line passing through the well, and that is parallel to the current flow direction. So when the current is set off, diffusion will begin from each of these spots. So by placing antiserum in a trench cut in agar parallel to the electrophoretic distribution of the antigen, the precipitating reaction can be observed and can be used to demonstrate the nature of diffusing molecules. So in this particular case, a broad band of antibody diffuses towards the antigen, form a linear antibody trench, while the antigens diffuse as expanding this. This results in a complex pattern of arc-shaped zone in the precipitate. So there are some advantages of disadvantages of precipitation reaction. So advantages are this is fairly sensitive and high specificity is there, and it's time consuming, and some costly instruments are required, some costly means high technical skills are required. These are the disadvantages. Some chemical bonds that are responsible for antigen antibody reactions. These bonds are electrostatic bond, hydrophobic interaction, hydrogen bonding, van der Waals interactions. That's all.